Thanks for the kind introduction and thanks for inviting me. Thank you to Dora and the organizing committee, of course. Thank you so much. So my name is Strand. I'm a professor at Uppsala University. Please interrupt if the sound is bad or if there are any problems with my slides and so on. I'll share my screen. I hope that works out well. So I'm at Uppsala University. I'm both at the Department of Information Technology and the Department of Surgical Sciences. My position is at the Department of Information Technology, but I have lots of collaboration with the radiology division at the Surgical Sciences Department. And what I will talk about today is a bit of general aspects of large cohort analysis in medical image processing, or big data kind of analysis in medical image processing, and some of the projects on whole body MR image analysis that I'm leading. And I'm also leading other projects on neuroimaging and radiation therapy with MR, DNAC equipment and so on. But this talk will not focus on them. So let's move on to the kind of background. The image processing development since the 60s or 70s up until today. So as you probably all know, that there has been a progress towards data-driven analysis since the 60s or 70s. And in the 70s, the image analysis and image processing was mainly based on low-level image processing, decision trees kind of algorithms. And this was partly due to lack of computational capacity, or to a large extent so, but also lack of image data. And during the years, the computational capacity and the image data has increased and is still increasing. So I would say that it's an exponential kind of increase for both of them. And the trend today is very clearly towards data-driven image analysis. So looser models and more focus on the actual data to do the analysis. So this is the way I would like to describe the situation we are in today. So the availability of image data is increasing exponentially. The computational capacity is increasing, and the method efficiency kind of hooks onto that and is also increasing. So what we have today is the deep learning kind of hype, as you all know. And this has the availability of image data and the computational capacity, and to enable computation on the kind of consumer hardware GPUs has made that possible. So to focus a bit on the computational capacity, I would like to say that the Moore's law is still in effect. I don't know if it's a law, but still, we see this exponential increase. So this is an image from Steve Jurvetson. So this shows the calculations per second per constant dollar over time. And the hardware, as you can see, perhaps now it's GPUs in the last few years, and this, as I said before, it's to a large extent actually driven by consumer components and gaming GPUs. Very efficient GPUs are available at reasonably low cost today. And if you focus a bit on the availability of image data, so for example, my cell phone, it's not a fancy one, but it has 4 cameras and a dozen apps that are kind of hungry to get me to acquire image data. So image data is acquired very much today compared to previous years, and this can be seen also in the kind of radiology and in the clinical setting. So here are some plots from a paper that I found that showed up until 2010. So it's actually a bit old now, but there is a dramatic increase in the amount of image data being collected, and this also puts high demands on radiologists. So in 2010, a radiologist needed to interpret an image every third to fourth second, all workday, every workday. And this has also been highlighted in the morning today by Leo. This situation with a huge amount of data in a clinical situation. And also research image cohorts or kind of biobanks, if you like, or data sets available. So cohorts here is a group of subjects who share a defining characteristic. What we can see is that there is lots and lots of image data available. So internationally, we have the TCIA, UK Biobank, where they aim for 100,000 whole body image scans and other image scans as well. So this is MR. There is a German national cohort, 30,000, and these are just some more or less random examples. There are many, many initiatives to acquire lots and lots of image data. And local cohorts in Uppsala, I mentioned them because I will show some examples on that in a while. So the POEM cohort, for example, where the imaging is done on roughly 500 subjects. So this is also whole body MR image data. And method efficiency. So how to illustrate that? We are all aware of the deep learning kind of era we are in now. I wanted to illustrate it in this way. So in 2012, we participated with an interactive method, but it's not a deep learning method, in a challenge, Mickey challenge, PROMISE. So this was a challenge about prostate segmentation. And we performed very well. We made it to the third place, and yeah, we did really good in this competition challenge. Today, so this challenge is still ongoing. Now we're at rank 245 out of 327. So it's a drastic decrease, of course, in the rank for our method. And what has happened since then? Well, the deep learning kind of explosion has happened. In 2012, that was the year when the AlexNet won the ImageNet challenge, the large-scale visual recognition challenge or competition. As you probably are aware of. So what we actually see today are methods that are based on kind of finding patterns in large amounts of data. So this is a data-driven approach with no or few predefined features. So we have seen a dramatic kind of development in the image processing methodology. All right, so what I wanted to show in terms of kind of projects today is it's two approaches for analysis of large-scale medical image data, and this is based on whole body image data, as I said, and it's magnetic resonance imaging. So one of them is called Imiomics, and the other one is a more recent initiative. So this is what we call aggregated saliency or cohort saliency. And built into the methods are, to different degree, assumptions, of course. So kind of formalized experts' experience and intuition. And we have the model that represents the assumptions by parameters to kind of different degree, and then the data is also very important here. We need lots of data for these methods. So let me start with the Imiomics approach. So this is something that we have developed. So this is an analysis of whole body image data and other biomarkers, and it's an approach where we from large cohorts with non-imaging data or kind of alternative biomarkers can do analysis without explicitly doing segmentation in the image data, but do this in a kind of holistic manner. I'll explain the concept in the upcoming slides, but what we can see here in a kind of traditional analysis, what we would do is to derive some explicit measurements from the image data. I don't know, the volume of the liver, for example, or so, and then do statistical analysis based on that. What we do in Imiomics is point-wise or voxel-wise analysis, statistical analysis. And it's kind of holistic because the whole human body is analyzed. So this is as opposed to region of interest-based method or segmentation-based methods, where information is extracted from predefined, pre-segmented regions. And all the collected image data is used in the analysis. And also this approach allows relationships to all other collected non-imaging patient information or subject information. So how do we do this? Well, it's based on image registration. What we do is we find point-to-point correspondences. So this is similar to what has been done in neuroimaging for several years, but we do it in the whole body, in a whole body imaging settings instead. So we have a correspondence between the same position for all of the subjects in the cohort. And with that information, we can do the statistical analysis voxel-wise. And in that way, we can get these kind of relationship maps or correlation maps or R maps, if you like, so that we can color code the correlation in each of the position in the human body. So it's essentially one statistical test per voxel. And the kind of key component here is image registration. We use volume images for this, but it's illustrated here on just a single coronal slice. And the image registration problem is difficult in this setting because there is a huge variability in anatomy or morphology in the human body, and for example, this subcutaneous fat repose varies a lot between subjects. So this is something that we have been working quite a lot on to develop. The imaging parameters that we focus on here, it's magnetic resonance imaging, as I said, it's fat content. So this is fat water separated magnetic resonance imaging, so MR images, with the Dixon technique, so that we, for each voxel, can get the fat content or fat percentage, so absolute values, which is really, really valuable in the image registration. And in the image registration process, we get a deformation field, so we can compute the local volume deformation in each voxel. And in some of the analysis, we also have access to PET data, or we also use PET data, typically FDG PET. So here's another illustration of the Imiomics concept. So we deform all the whole body volume images to the same reference space, so the same coordinate system. And once that is done, we have the point-wise or voxel-wise correspondences, and we can do the correlation analysis. And in that way, we can get these colorful correlation maps where each voxel is color-coded with a R value or correlation to the non-imaging data or the biomarker that we're interested in here. We will look at a number of examples in a short while. Method development in Imiomics. We have developed an image registration method, or there are actually a few different versions of this, where we utilize tissue-specific elasticity constraints. So we make an assumption that bone is relatively rigid and has low inter-subject variation, and that lean tissue is less rigid and has higher inter-individual variation, whereas adipose tissue or fat is highly elastic and has high inter-subject variation. As I said, we map all the subjects or the whole body volume images to the same coordinate system, to a reference coordinate system. And what we have done in some of the analysis is to just pick one subject and decide that this is the average reference coordinate system. But what we ideally want to do is to actually compute the average reference space. as an optimization of the reference space. So we have developed a method to generate the reference space for the whole body image processing that has 0 voxel-wise average volume change when mapped to a cohort. So if we map all the whole body volumes to this computed average reference space, then the average deformation is 0. We have also developed methods for multiple comparison correction. As I said before, we do voxel-wise correlations, statistical tests, and of course neighboring voxels are not independent. So there is a dependence between neighboring voxels. So then therefore we have developed multiple comparison correction techniques. Methods based on permutation-based cluster extent correction and threshold-free cluster enhancements perform very well in this. But there are several different approaches for that. Bonferroni is a bit too restrictive because we're doing so many statistical tests. Let's look at an example. So this is a proof of principle analysis, correlations between local tissue volume and the total fat mass, lean mass, and also correlations to waist-hip ratio. So the image data here, this is the POEM cohort, the local Uppsala cohort. Lars Lind is the PI. All the subjects are age 50, and in this analysis, we use 326 subjects. All right, so the first analysis, this is tissue volume, local expansion contraction in the image registration. So this is tissue volume, and then point-wise correlations to the total fat mass, how much fat mass the subject has in total. So this is measured by bioimpedance analysis. And here we can see, again, this is a proof of concept. So we can see that there is a positive correlation between total fat mass and subcutaneous fat. So this is in female, coronal slice. Also to the size of the liver, and also perirenal fat. I don't know if we can see the kidneys. It's a bit difficult, but that is the case. And a negative correlation in lungs. So the more the total fat mass, the smaller the lungs. And then we also did in this proof of concept analysis tissue volume versus lean mass. So then we can see that the more lean mass the subject has, the more muscle it has, which is expected. So the lean mass, that is essentially muscles. We also see a positive correlation in the long bones in legs and arms. You see the red color here. Positive correlations in lungs and also in liver. So what I'm showing here is a coronal slice. It's actually a curved coronal slice visualized to the right here, and then a number of axial slices also shown in this illustration. Positive correlations are color-coded with one color, so red is strong positive correlation, and negative correlations are color-coded with cold colors, so blue is the strong negative correlation. Waist-hip ratio, we can see as expected that we have a positive correlation to the subcutaneous fat around the waist, and it's also a positive correlation in the liver, negative in the lungs again. So the imaging biomarker that we use here, that's tissue volume. We can also use fat content. This is information that we have from the fat water separated MR images. So this is also point-wise correlations. So fat content versus total fat mass. And here the correlations are a bit weaker, but we can see a positive correlation in liver, for example. And here we can see the perirenal fat more clearly. So positive correlations in liver, meaning that the higher the total fat mass, the higher the fat content in the liver. So this is proof of concept analysis. Let's look at a more realistic and perhaps interesting from medical point of view analysis, Imiomics analysis. So this is Imiomics in the metabolic syndrome. And the metabolic syndrome is the medical term for a combination of diabetes, high blood pressure, and obesity. If you have the metabolic syndrome, it puts you at a greater risk of getting coronary heart disease, stroke, and other conditions that affect the blood vessels. So it's a combination of the increased abdominal circumference. So this is if you have abdominal obesity. Here it's called waist circumference. Right, so what we see here is that if you have high waist circumference, then you have more subcutaneous fat, also more visceral fat. We see negative correlation in the lungs again. This is again now in the POEM cohort. The colors are coded a bit different in this analysis, but in a similar fashion. So this is actually based on group comparisons instead of correlation analysis, but that doesn't matter that much here. And then another parameter or criteria for the metabolic syndrome is the HDL cholesterol. So elevated, so this is HDL cholesterol. And here we can see a positive correlation in the liver. Fasting glucose, so this relates to diabetes, also positive correlation in the liver. Blood pressure is also measured as one component of criteria for the metabolic syndrome. And here we can see a positive correlation or positive relation in the upper body, the subcutaneous fat, the liver, and so on. So this is tissue volume all the time. And then we also have triglycerides, so blood fat essentially from circulating biomarkers. We see a positive relation in the liver and also in the subcutaneous fat in the upper body. These are female subjects. So if we add all of these together, waist circumference, HDL cholesterol, fasting glucose, blood pressure, and triglycerids, we can get the metabolic syndrome. So the criterion is that you should have if you have abdominal obesity and two other criteria, then you have the metabolic syndrome. And here we can see the correlations with the metabolic syndrome, so the relations to the metabolic syndrome. And this analysis gives understanding on how the different components actually add to the full metabolic syndrome. Let's move on to another application. So this is anomaly detection. So in Imiomics, we do a representation of normality. We have a whole body atlas. And if we do that on normal subjects, then by doing pointwise statistical tests of a new subject, we can see if there are any anomalies from the expected or from the normal material. And this is based on PET data. So the registration is done by the MR data, but then we have PET data for this as well. And the normalization of PET data was done by mediastinal blood pool mean FTD uptake. And so this is normalized PET data. The criterion is a large region of high FTD uptake. So this is in another cohort. And then we can see that we can actually detect anomalies automatically with this technique. And then for the Imiomics analysis, we have applied this to several other non-imaging biomarkers relating to obesity and diabetes, and we're also doing genetic analysis now, etc. Let's move on to the second kind of cohort analysis or large-scale data analysis. So this is aggregated saliency, and this we applied to the UK Biobank data, and this is a regression technique. So what we essentially did was to do regression where the independent variables is the image data and the dependent variable is something else. In this example, it's age, but we will see examples of other dependent variables, liver fat and body composition markers, etc. So what we did here is we implemented a regression task. So this we have used both BDD and ResNet architectures. So from a large cohort, predict for each subject a single outcome variable. So this is the regression. And then we try to train the network to estimate this single outcome variable as well as possible. This is an example on prediction in years. So this is the age, the prediction versus the ground truth. You can see that we get quite good prediction of the age. So this is a basic task, and on top of that, we added aggregated saliency. So this is kind of part of explainable AI, or if you like, this saliency mapping. So we did that for each subject, saliency maps by GradCam, then gradient classification maps. And then we added all of the saliency maps to a single output. So this figure illustrates this. We have a two-dimensional input. We project the saliency to a three-dimensional space. We deform it to the reference space again, and then we project the saliency to the reference space. And this is the kind of result that we have achieved. So this is a preliminary study with 3,000 subjects in the UK Biobank imaging cohort. We have also results now on bigger data sets. So this is what we can see in the aggregated saliency when it comes to visual adipose tissue. We can see that the visual adipose tissue is actually highlighted. DEXA, so this is the total fat mass. Left thigh, we can see that the left thigh is actually lit up here, and right thigh, likewise. Liver fat, BMI, here we saw that the knees and the lungs really seem to be salient for this feature. Pulse rate, then we get response around the heart, and grip strength. So we see a strong response in muscles and actually also lungs. So this is also kind of proof of concept analysis, but we can see that it actually seems to work. Okay, I'm at the conclusions. So we are today in a unique situation with access to so much computing power, algorithms, and image data. I didn't talk that much about AI, but many believe that computer-assisted methods will revolutionize the clinical workflow. And in some narrow tasks, computer-assisted methods perform better than human. Imiomics, aggregated saliency, and other computer-assisted image processing methods can find patterns in the huge amount of data available today as a complement to human experts. And this enables increased disease understanding and automatic disease and pathology detection, etc. So this is my last slide with acknowledgements. Thank you so much, all.
Okay, thanks for Professor Robin's excellent presentation. And I will, because due to the limited time, we can have only one question from our audience. So do you have any questions? You can either directly unmute and ask the question, or you can leave the question in the Q&A or in the chat window. Okay, Professor, may I ask you a question? Because I remember you mentioned the metabolic syndrome. So I just want to know, since the metabolic syndrome is sometimes a prodromal case for the diabetes, so do you think your research can help contribute, like contain, let's say, the diabetes and help the treatment?
And help the treatment. So this is a cohort analysis to find patterns in large data sets, so to speak. But this can for sure generate understanding of the disease. And I also want to mention that we have done advanced analysis when it comes to diabetes biomarkers. So we have analysis in that direction with which actually increases the understanding of the diabetes. Direct analysis for the kind of diagnosis. We're not there yet with this technique, but then again, it's more a tool for generating disease understanding.
Okay.